Just want to make sure we have time for our last two presenters. Representing the Bachelor of Science degree in Psychology, we have Anna Lysenko.
And I'm going to be presenting research that I did over at the Institute for Basic Research on Staten Island. It's a phenomenal facility which focuses mainly on mental disabilities and disorders that come along with brain damage and whatnot. My main focus was on Alzheimer's disease. I worked in their neurobiology department under my phenomenal mentor, Dr. Janusz Verkowiak, who helped me so much in everything that I've learned. And so I want to start off by pointing out a really important fact that the problem with Alzheimer's disease is, sure, there are many ways to diagnose it, sure, there are many ways to identify it, but there's no surefire way to put like an official medical diagnosis on the papers until postmortem autopsy reports are performed. So because of the need to identify very specific biological markers that come along with the disease, autopsy reports are performed when the person is deceased, and they specifically look for amyloid plaques and neurofibrillary tangles. There are a few assessments that you can do while the person is still living, although this is not a guarantee that the person is suffering from Alzheimer's disease. The main thing that you look at is psychological symptoms, dementia, loss of memory function, cognitive decline, not remembering where you're going, not remembering what you're doing, just the simple things that we can observe every day that you forget. Another common test is done through a spinal tap, and they test for t-tau, p-tau, and amyloid beta 42 protein levels. So in terms of these, they're not specific to Alzheimer's disease, although they are extremely common, which is why they do test it. T-tau and p-tau levels are increased, while the alpha beta 42 levels are decreased. So it's a pretty solid way to test if you go along with the psychological symptoms, but it's, again, not a guarantee because it can be associated with a number of other diseases. My research, mainly, we focused on the brain development of mice. So everything that I will be showing here is done in a mouse model. We did immunostaining, and we worked with a confocal microscope, and the specific immunostaining antibodies that we used for our immunostaining were Ki67, which is a stain for proliferating cells, and double cortin, which is a stain for immature neurons. Up here you can see a picture. This is the hippocampus in a mouse brain. It's specifically the CA3, which is the corneomonas 3 section of the brain, and the dentate gyrus. You see all the bright green staining up there. Those are all immature cells and proliferating neurons, and this is in a control mouse, so you see very, very gorgeous staining. You see a lot of cells. You see a lot going on here. But if you look at a transgenic mouse, it's kind of plain, and you don't see any of the bright green. So this is very, very common. You do not see any, you see very little, if any, immature neurons going on in the sections of the transgenic mice. We also do immunostaining for light microscopy, and that's mainly to identify the amyloid plaques that build up, it gives us a very, very clear indication, and you see the changes as the time goes on. So you start off with very, very little in, say, about 3 months, and then you go on to a lot more as the mouse progresses and grows. So this is an example of a 3-month-old mouse. All the brown staining that you see across is, those are all the amyloid plaques. So there's a lot, but not that many. If you see up toward the top that it curves a little bit, that's the hippocampus and the dentate gyrus, which is what we focus on in terms of the proliferating neurons. And this is the hippocampus in a 28-month-old mouse. Now, 28 months is a really long time for a mouse to live who is transgenic, who does have Alzheimer's disease, or the biological markers of the disease. So we see an abundance of plaques here. They're very, very clear. I also wanted to point out that a mouse cannot naturally develop these biological symptoms of Alzheimer's disease, or the psychological symptoms, obviously behavioral tests. But they create these transgenic mice who overexpress human genes that they think are really associated with Alzheimer's disease, and through that we're able to get the symptoms that humans will have. The major neuronal differences that you see in terms of control mice and the transgenic mice is obviously the amount of proliferating cells and immature neurons. The data is currently being analyzed. We're kind of in the middle of the statistical analyses of the project, so it's not complete. So I couldn't tell you if there is a statistically significant difference, but based on all the pictures that we've studied, we've gone through hundreds of mice and hundreds of slides and sections, and it's pretty obvious that the control mice have significantly larger number of these stained neurons than the control mice do. We do behavioral testing on our mice as well to see if they have symptoms that humans possess, like cognitive decline, loss of memory function, whatnot. A very common test that is done is called a wet maze. I have a picture of it. It's this. It's a big tub of water, and what we do is we put the mice in the water, who are phenomenal swimmers, in case you didn't know that. The water is opaque, so it's dyed with a paint. It's silicone paint that's not harmful to the mouse at all. So the water is opaque so that they can't see what's going on underneath the water. And when the mouse is put in there, there's a marker on the side of the mouse that helps the mouse identify in which direction there's a platform. There's a platform right off to the side a little bit, sometimes in the middle, that they let the mouse swim around and find it while it's out in the open so that the mouse can see what it's swimming toward. And after a few shots and after a few rounds of doing this, the platform is lowered so that it's underneath the water, and the mouse should be able to identify this marker, whether it's on a wall or in the tub, and recognize where the platform should be. So even when the platform is hidden, the mouse should be able to find it after training and after a few runs of it. What we see is with the control mice, they do it very quickly. They learn it, and after a while, instead of just swimming around all over the place, they just go directly toward the platform to get out of the water. But with the transgenics, who, especially the older ones, who are put in there, no matter how many times you do it, they do not find the platform. They have trouble identifying the marker. They have trouble figuring out where anything is. You see this through other tests that we do, such as Pavlovian fear conditioning, in which you administer a beep and a shock and see if the mouse can associate the two through freezing after the beep. And when the shock is administered, the mouse usually jumps but is prepared for it in the control mice, but the transgenics have a significantly harder time learning. So basically we run a lot of tests on these animals, and they do portray a significant amount of the human symptoms that we see in people who suffer from this disease, and they're just trying to figure out the differences in the cells and in the neurons and in the behavioral tests and see how they can connect all of that. And that's it. Any questions? Yes.
What's a transgenic mouse?
Well, they breed, I'm not exactly clear on what they do, but I know that they breed these mice. It's also common to do knock-in, knockout mice who either lack a gene or have an extra, like something that another mouse that they're testing does not or has it, or so they breed these mice, and I believe that it's done intrauterine. They somehow can extract genes or whatnot, or overexpress genes. I know there's a really common gene for Alzheimer's disease. It's the E4 allele of the APO gene. I don't know if they use that to create these mice, but they somehow get genetic factors that are overexpressed and help us produce these transgenic mice who have these symptoms. Yep.
Do normal mice also develop these Alzheimer's-like brain structures?
No. No, amyloid plaques and neurofibrillary tangles are not something that a regular mouse can develop. That's it.
Thank you.